Welcome to Marine Science Day. I am so excited. I hope you're having an amazing time. I know that all of our researchers are so excited that you are back and that we are able to show off all that we've been doing to all of you. So thank you for being here. This is a hybrid event. So that means that there are folks that are also joining us online as well as the folks that are in the room. So for the folks that are online, if you have any technical issues, Shannon is our volunteer. Hi, Shannon. You can put any technical questions into the chat and she will help you work through that. For those folks that are online, if you have questions for today's speaker, also put those into the chat and we'll work through them at the end of today's presentation. For the folks in the room, thank you for being here. Again, because this is a hybrid event, that means that to ask questions, you will need a mic. So when we get to the question and answer section, if you raise your hand, I'll either bring you this mic, you guys get to watch me run all over the place, or you can go to the mic stand that's right there. But that allows our folks that are online to hear the questions as well. Wanted to make one quick announcement for all of you. If you like these kind of events and you want to keep learning, we do a Science on Tap every single month. It is an evening event that's open to the public where we highlight an in-depth research project. This month, we are building on this Marine Science Day topic. We have several researchers that are going to be talking about the sea urchins and their population impacts on the coastal kelp forests here in Oregon. So if you're interested in that event, that is April 26th. And if you want to learn more about anything that Hatfield does, if you go to the HMSC website, you go to events page, you can find out all the information you need to know about how to log in there to get information about where the events are going to happen. But without further ado, I want to start us on today's keynote speaker. I want to do a quick introduction of our speaker. We have Dr. Sarah Gravem here with us. Hi, Sarah. Sarah is, yay! Sarah is a research associate with Oregon State University's Integrative Biology and PISCO group. PISCO stands for the Partnership of Interdisciplinary Studies of Coastal Oceans. She is a marine ecologist that works at the intersection of community and behavioral ecology. Dr. Gravem focuses on rocky intertidal kelp forest ecosystems and looking at how species interact with communities and how those are in fact impacted by climate change. She is particularly interested in how predators affect prey populations and prey behavior and the resulting consequences of those key interactions to the rest of the players in the ecosystem. And we are thrilled to have Sarah here today. So I'm going to hand this off to Sarah. Go ahead and turn on your mic. I'm going to turn the lights down.
Okay, can everyone hear me? Okay, on the mic even? Okay, sounds great. Okay, thank you, Cinnamon. Thank you for coming. Marine Science Day is back. And thanks for having me. So my name is Sarah Gravem. I am a research associate at Oregon State University. I work here at Hatfield a bit. I'm part of the PISCO group, which she already said the whole long word, so I'm not going to say it again. I also am the founder of the Primed Network, which we'll get into later, and a member of the Oregon Kelp Alliance, which we will also get into later. I'll be talking today about the theme of the entire conference, right? So connecting our community above, which is us, to our community below, which is ocean critters and the ocean ecosystem. We'll be focusing on two ecosystems. First, we'll dive into the rocky intertidal, and then we'll go sub-level and we'll get into kelp forests. But first, we're going to play a game. And the game is Name That Critter. And so if you know the name of the critter, you should shout it out. And I might ask you to elaborate. Okay, you ready? Okay, don't be shy. It hates me. Okay. We're doing it that way. Okay. Crab. This is specifically a cryptic kelp crab. Why is it called cryptic? What's cryptic mean? Hidden. Why is it, what is it hiding in? Under? Close. Algae, right? There's some pink algae on its head. That is not part of the crab. It actually plucked it and stuck it there a little decoration so that it can what? Why is that a good idea? It's hiding from its predators, right? So it doesn't want to get chomped up by a seal or a sea otter. What about this? Chitin. How do you know? Plates. How many plates? 8 plates. Every time you see something with 8 plates on its back, it's a chitin. And chitins, are they really fast? No, they're so slow. They're so slow. Like you could watch it all day and it wouldn't move. They eat algae stuck to the rocks, right? They are not predators, you guys. They're way too slow to be predators. They just they scrape the rocks for algae. This is a line chitin, which I think is the prettiest of chitins, so it's my favorite. All right, Heckler, this is my son. All right, what's this? What's this? Isopod. It's kind of like a bug. It's kind of like a roly-poly or pill bug that you see, right? But this one is a marine isopod. And I want you guys to all check out those claws that it has. Check out those sharp, sharp claws. Next time you go to a tide pool, if you ever see one of these, try and just gently tug on it. Gently. Don't squish it, but gently tug on it. That sucker is not coming off. They are so hard to get off because of those sharp, sharp claws. What do you think? Why do you think it does that? Why does it stick to algae? Elliot. Yes, it has a nice strong back and it holds on tight so fish can't snatch it and eat it. You're right. Excellent. That's an isopod. Okay, what is this? Sea anemone. Did anyone touch a sea anemone today? Cool. What did it feel like? They cheated. They cheated. It's sticky. And when it's when you touch it and it sticks to you, what's happening, Boone? It's shooting thousands of microscopic harpoons into you and it's trying to poison you. But we're too big. If we were an isopod, on the other hand, we'd be like, ah! And we'd be stuck in that thing and we'd be goners, right? But because we're big and we have thick skin, we just go, it's sticky. So this is a red beaded sea anemone. This is around here. And this is on my shirt. Sea slug. What's another word for a sea slug? Nudibranch, right? It means naked gills, which is nice and fun. This is the type of nudibranch or branch. I don't know. There's no right way. Does anyone know what type it is? Yeah, Jake knows. It's an opalescent nudibranch, and it eats things kind of like the sea anemones that we saw in the last picture. And what did sea anemones have in their tentacles again? Harpoons, right? What do you think this nudibranch has in its serrata, which are hanging off of its back? Harpoons. It does not make the harpoons. It eats the harpoons in the hydroids or sea anemones that it eats, and it has a sweet, sweet trick. It eats the whole cell without getting stabbed in the mouth. It digests it, it goes through its guts and up into its serrata, and it puts it up there. And then when a fish comes by, the fish gets harpooned. Go away, fish. No fish, no nudibranch for you. So that's why they're able to be so pretty and colorful and beautiful, right? Because they're super toxic and scary and no fish want to eat them. This is an opalescent nudibranch. What's this? Sea spider, right? And if you had to make a guess, which end's the butt? Which side? Point. This end. No? This end? You're right. You guys are so sneaky. Okay, so this end is the butt, unlike spider butts on land, right? That end is the mouth. And you can see the two tiny little brown eyes up there, right? That's its mouth. And this big old honking thing, that's its proboscis, which it sticks into sea anemones and slurps up. Just kind of like spiders slurp up insects after they bite them. These guys don't make webs. They just kind of crawl around the ocean and go. The anemones can't move, so, you know, they can't do anything about it. Okay, this is not an animal. What is it? Close. Seaweed. It's seaweed. It's green seaweed, and it's called dead man's fingers, which is a great name. Hey, Cal, do you want to eat dead man's fingers? No? You do? Yeah. Who would eat dead man's fingers? I would. They're actually pretty good. People put them on burgers and stuff. There's a whole YouTube channel about eating dead man's fingers. I swear. Go check it out. Called Black Forager. Do it. Okay, this is a tricky one. This is not an animal. It's evidence of an animal. Good guess, Theo. It is sea snails. Can everyone hear me in the microphone, or is it just going in and out? Okay. Yeah, I'll just yell. All right, so this is evidence of snails. You're exactly right, Theo. So this is the tracks that a snail makes as it grazes over algae. So you can see those little white scrapes, right? It has a tongue called a radula with scrapey bits on it, and it rolls around the intertidal and like lick, and that's why it's zigzag. They actually like mow the algae, and then they leave slime behind. And guess what algae likes to grow on? Slime. So they farm. They make their own little algae farms, and they leave behind fertilizer, and they leave it for a few days to grow, and then they come back and they mow it again. Isn't that cool? So you can see the old tracks and the new tracks right there. Okay, this is the last one. You better know this one, son. Sea star. Okay, sea star. This sea star, does anyone know what the species name is, or the genus name? Jasper. He was my student. Jasper knows because he spent a lot of time analyzing photographs. This is Pisastrocracis, and this is the sea star that brought me to Oregon. So I came here after my PhD to do research. And I came because of what we like to call the Pisaster disaster. And as probably many of you might remember, there was a disease event that swept through back in 1314. And what would happen is the sea stars would get these little lesions on their bodies, little white spots, and then their arms would fall off. And then the arms would crawl away like zombies. And the sea stars would end up melting into a puddle of goopy goo. And this happened over a couple of days. So this is a one-day time lapse that we took. And it was really horrible and very sad. I was pregnant with the loud one. And I actually had just had him, and my student calls me and she's like, "Oh, your sea stars are dying." And I'm like, "You're fine. Go look again. You're full of it." And nope, she was right. All the sea stars were dying. And so that is when my life changed a lot, in the fact that I had a kid, and also this happened, which completely changed the trajectory of my research career, because that became the sea star wasting disease outbreak. So it started in California, where I was, right around here at the time, and in Washington in 2013, at the same place, but totally, or same time, but totally different places. Notice Oregon got skipped at first, which is super weird, right? And it spread over the next 6 months or 8 months, all along the coastline. And it eventually went from Baja to Alaska to the Aleutian Islands. Huge, huge expanse. And it affected over 20 species, we think. And we now know it's the largest marine epidemic on record. So we think it was caused by a virus. There's been some debate about that, but best evidence right now is still virus. They're testing it, but no definitive answer there yet. It's still happening. You can still go out and see sea stars that are infected. It's not like epidemic proportions like it was, but it still lingers around. And the reason that we are able to make maps like this, and the reason we were able to track this event so well and with so much detail over such a huge amount of area, is because of people like you. So thousands of you, has anyone submitted a sea star observation to seastarwasting.org in this room? Thank you. These are this is a website that we have. It's a community-based open source website. Anybody can report to it. And if you see a sick sea star or even a healthy sea star of any kind, you can take a photo of it, record your location, fill out the little form, and send your species to us. So I'm going to have my trusty assistants each go to a corner of a room, and you're going to hand out your bookmarks. And if you see a sick sea star, you're going to send it to the website that's on that bookmark, okay? If you don't want one, that's fine. And if there are any leftovers, you can just leave them on the aisles, and the kids will come and get them later. Thank you, gentlemen. All right. So the two species that were hit the hardest, or at least very hard, by this disease are Pisaster ocratius, the Pisaster disaster. That's the one that many of you probably touched today, or at least have touched in the Oregon intertidal before. But the second one we'll talk about is the sunflower sea star. Has anyone touched a sunflower sea star? Not that many of you. Why? Only my students and my kid. Huh? They're super rare. They're not around in Oregon anymore. They used to be all over the place, and they're not here anymore. We're going to talk more about that in a little bit, but keep the sunflower sea star in your mind. So, pop quiz. What do Pisaster eat? Somebody except for Boone. Mussels. Thank you. They eat mussels, they eat barnacles, they eat snails, they eat all kinds of stuff. And they actually eat by capturing it, and they barf up their own stomachs and let their stomachs out like bleh, and then they just leave their stomachs out for a while, digest, hang out for a day, and then they slurp it back up and go find another one. How gross is that? They have terrible manners. But they're really important top predators. You wouldn't think they were scary just by looking at them, right? They're kind of cute, kind of slow, but they're actually the wolves or the lions of the intertidal zone. They're the top predator. And they're so important that they can actually create what their ecosystem looks like. They actually cause intertidal zones. So when you go to the shore and you walk from the high intertidal down to the lowest spot you can go, you start out in like barnacles and some scuzzy looking algae, and then you walk over a mussel bed, and then you walk down into the slippery algae, and then you're like, oh God, oh God, oh God, and then you fall on your butt because there is algae everywhere. Those zones are caused first by the environment, but secondly by predators. And the bottom of the mussel bed zone is controlled by these sea stars. So all these sea stars, this is the sea star buffet, right? They're eating these mussels and scarfing them down, and the mussels are stuck high up on the shore where the sea stars can't get to them. I know. Can people in the back hear me okay? Okay. Maybe I'll just turn this thing off. Oh no, I can't. Sorry, I can't. All right. So under normal circumstances, when this happy little sea star is futzing around in the intertidal, it's chomping up mussels, and by keeping the mussels up high, there's all sorts of space for everything else to live, right? Chitons that we just saw, the anemones that we just saw, the algae that we just saw, the dead man's fingers, they all have places to live. But when you have, so with the sea stars, that means there's more species, higher biodiversity. And back in the 60s, one of my advisor's advisor, so my academic grandfather, took and removed all the sea stars off of one little stretch of coast and chucked them sideways into the ocean for 10 years. And this is what happened. the mussels went and took over everything and moved down, and the biodiversity of the system went down. So that was actually one of the first experiments ever to show that predators have actual consequences and benefits to their ecosystems. Before, we'd just be like, it's a wolf, shoot it. Ah! And after this was when we really transitioned into appreciating what top predators can do. So when this wasting disease happened, we were like real sad, of course, for a hot minute. But also it was like, oh, whoa. Because what do you do if you want to study something and understand its effects? You take it away, right? So nature came in and took this one important top predator away within a matter of months over its entire range, which, sad, but scientists dream, honestly, as far as like understanding what it does for its ecosystem. So that's what changed my life, and that's why I came to Oregon. So my research questions broadly have to do with how this disease has affected intertidal communities, so I can understand how important that predator is to its ecosystem and how important all predators are to all ecosystems. So our main questions right now for today anyway are, have these mussels increased? Because we know these sea stars at the top eat all sorts of stuff, but their favorite food is mussels. So when the sea stars die, we should see mussels increase and all the other stuff go down. So our next question becomes, has diversity declined? And then, of course, we love our little colorful buggers. And so we want to know if the sea stars have come back. Okay, so this begot the STARS project, which is called the Sea Star Tragedy and Recovery Study. Very fun. And it is 33 sites from just north of here near Cascade Head, all the way down to Southern California, south of Disneyland, right? And we track these every year since 2014. And I've been in the car a lot. And this is a really big collaborative effort, and that's what PISCO is all about, between Oregon State, UC Santa Cruz, and Concordia University, which is down in Southern California. And another big piece of this puzzle is people like you. Has anyone been to a BioBlitz on the Oregon coast? Yeah, so if you haven't been to a BioBlitz, go to the BioBlitz this year. Oregon Coast Aquarium hosts them every summer, usually in July-ish. And we usually partner with them to actually collect real data using people like yourselves. So we'll take folks out and we'll say, here's this transect line. We're going to lay this down. And hey, Karen, get on your hands and knees and look under that rock for 10 minutes and see if you can yank off all the sea stars you can find, because we're going to try and track how big they are and how many there are, so that we do this every year and we can see how fast they grow, how fast they come back. So this is an example of what we do at these sites. It's not just sea star surveys. We do experiments. We take water samples. We monitor the community. We take pictures, all sorts of stuff. So a bunch of the students in this room are these people in this shot of a time lapse of us just busy bees working one tide. And we do this almost every single low tide with good weather, every year, all year, for decades. So this is what we end up with when we track these communities. So this is a shot of a plot actually down near Santa Barbara. It's on the military base down in Vandenberg, which it's really cool to go to because you can see the spaceships as you go to the intertidal sites, which is great. No one else can go there. You have to get a military ID. Bonus. So in this plot, we have some mussels right here in this mussel bed. And this is back in 2018. So this is just after the disease, not too long after. And we have some barnacles. Oh, my mouse is gone. There's some barnacles and some green algae and a little piece of kelp and a bunch, all that little scrubby, sandy looking stuff is sea anemones. So that's 2018. And then we went back the next year and you can see what happened. Barnacles are gone. More mussels. What else? Less sandy stuff. I think there's a little more sandy stuff. There's definitely less algae and less kelp, huh? Right. And the barnacles got bigger, if you noticed, but there's fewer of them. Okay, what happened now? Yeah, they're taking over because they're bullies. They're the bullies of the intertidal. They come over and they're like, hey, get out of my way, kid. It's my spot. And so they kick everybody else out and they take over. These are all little anemones over here. So we went from like 5, 6 species down to 2. All right, so let's look at what this data looks like over time. So that plot I just showed you ends up being put into these dots on this graph. And we can look at what's happened in these ecosystems from 2015 to 2020. And mussel abundance is here. So this is like total mussel cover, 100%, and this is 0. And what can you see from this graph? Are these lines going up or down? Up. They're all going up. Mussels are going up, 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 up everywhere. Or they're flat. But nobody's going down, right? Why is that again? The sea stars are gone. Right. So one thing to note too is that the lines are all different sites. So we start with purple up by Cascade Head, north of here. This Cape Foulweather's just right over here, Depot Bay. And all the way down here, that's like near Disneyland. So what you can see is that actually the blue ones are going up faster and the red ones are going up faster. So Southern California and Oregon is where the mussels are going bananas. And in Central California in the orange and Northern California in the green, they're going up, but not very fast. Okay, so we know the mussels are increasing. Jasper, shout out. He analyzed a lot of these photos. Good job, Jasper. And then probably more of you. And so the other thing we want to know is, has biodiversity declined, just like that photo I showed you. So we are seeing generally biodiversity declines. So these slides are mostly going down, but it's not necessarily everywhere. But it is strongest in Oregon. And that's where we saw the mussels go up the most, right? So we're seeing a general biodiversity decline, especially in Oregon. And that suggests that the sea stars are the most important to their ecosystems, have most consequential effects on their ecosystems in Oregon and in Southern California. But Northern California and Central California are more resilient to community change, or resilient to what we call a trophic downgrade. So the top of the food chain getting cut off. And so our biggest questions now revolve around what makes an ecosystem stable, right? Because there's places all over the world where predators are being hunted or extirpated or their habitats destroyed. And we want to know, what are the pieces of a puzzle that make it more tough in the face of change, or less tough in the face of change? Okay, but everyone really wants to know, are the sea stars going to come back? Are they okay? And so the good news is that they're doing pretty well here in Oregon. So this is the Oregon panel, and you'll see 2010 to 2021. And the densities of sea stars are here. The adults are in orange, so that's that color. The juveniles are in purple, and the recruits, the teeny tiny babies, are in blue. And what you can see first is that before the outbreak, which is this red line, the adults were jamming around, super abundant, and there weren't a lot of babies or juveniles. And then when sea star wasting hit, we saw this big dip, right? It declined a bunch, and they stayed there for a little while. But I don't know whether we got lucky or whether the disease and the recruitment were linked to each other somehow, but they went out with a bang, you all. They had a big party on their way out. And a year after sea star wasting disease hit, the babies came. And they are so cute. They look just like this. But you see this big, big spike. That is a bigger spike than we had seen in 25, 30 years. And those little babies came in. They're actually still trickling in. And they, over the next couple years, right, started growing. So we then see the juveniles ramping up. And then over the next couple of years, those start growing, and we see the adults ramping up. And so now we're actually back in Oregon anyway, and have overshot how many sea stars there used to be. There's more sea stars now in Oregon than there were before sea star wasting disease, which is crazy. They are smaller. So like the biomass hasn't recovered yet, but they are doing great and they're on their way back to being normal again. The story is not so great down in California. So California had some patchy recovery and some patchy recruitment. And we don't have great data beforehand, and we're working on it. But what you can see is that like there's some babies, there's some adults, but it's not like nuts like it used to be. And it doesn't look like they're increasing too much anymore. That's in Northern California. In Southern California, I can tell you, like, we'll go to a site and we'll be like, there are 2 here, guys. Like, that's how few there are. There's one place in Santa Barbara that there's been 1 under 1 rock for 10 years, and that's it. Poor guy, lonesome George. But in Oregon, because we're Oregonians, things are going great. This is what Cape Perpetua looks like right now. They are just having a great time, and they're just eating, right? They're buffet, snack time for these sea stars. So we've got a really rosy picture for Oregon, but let's go a little deeper. And it's getting a little sad for a second. So does anyone know what this is? We talked about it already. Sunflower sea star. How big do they get? Like this? Like this? Like this? Like this? Yes, like this. And they have 20 arms, and they are one of the fastest sea stars in the world. They book. They are predators, and they are scary, and I don't want to be a clam in their way. But they got hit really hard by that wasting disease, to the point where a couple years after wasting, when those ochre stars started coming back, we were like, come on, come back. Nothing. And so we got really concerned. And a couple years ago, convened the Pycnopodia Recovery Working Group. And our first order of business was to do a global sunflower sea star analysis. So I and colleagues gathered data from all the data we could find in the whole world about these things. And this is their range. They go all the way to the Aleutian Islands and to Mexico. We worked with people from 3 countries and Canadian First Nations. We broke the data into 12 regions. 67 different people contributed data, and we gathered 31 data sets that span 53 years and over 61,000 surveys. And our first question was, how severe was this decline? So bad news is that from Mexico to the coast of Washington, it's like over 97%. And it has not changed in any recent times. So it's essentially they're gone in most of that stretch. And most of those couple were from like 17, 18, and they're not there anymore. We've seen 1 Pycnopodia in California in 5 years, like 1. It's crazy. I'll show you it in a minute. And then back up in, so if we get into Salish Sea, it gets a little better. It starts to get like 92. So low 90s on the Southeast Alaskan coast and the Gulf of Alaska. And then out in the Aleutians, the data are a little thin, honestly, but we think they did okay out there, like only 60% declines, right? But there are patches of them left. And then we also wanted to know, what did the crash look like? How steep was it, and when did it happen? So what you can see on this one is the timeline again, so 2012 to 2020. And we're trying to figure out when it hit. So what you can first see, each dot is what we define as crash day. And the reds and oranges are California, and the green is Oregon. And the x-axis is the probability of seeing a Pycnopodia on a survey. So up at 1 is like, if I go on a dive, I'm gonna see one. And down here at 0 is, if I go on a dive, I'm definitely not going to see one. And so what we have is, you used to be able to go on dives for the most part in California and the orange here, and mostly in Oregon, and be able to see one. And that dropped quick over those years and has flattened out to 0, which is sad. And then in Alaska and British Columbia, all in the purples, you can you used to be able to definitely see one, and now you're going to see one like maybe half the time. So, but what's interesting about this graph is how pretty it is and how rainbowy it is, you guys, because that means that from north to south, we actually have crash times that go south first, and then it just marches north, right? And that tells us a lot about how the disease worked. So there's faster, steeper declines in the south. And our next question then was how many died? So this is our estimated population size. We actually multiplied up like the number of surface area and the habitat and the density and all sorts of stuff to get like an actual population size. So this is in the billions right here. And what you can see is back in 2007, 2013, it just popped around in the many billions. And then 14 hits, which is when California got hit, and it drops. And then 2014, Oregon gets hit, drops. 2017, Alaska and the Aleutians get hit, and BC gets hit, drops. And now we're down in this density, which is a fraction of what they had. So it's a around 90% decline globally. We think over 5.75 billion animals died, and there used to be over 6 billion. So that actually qualified them for IUCN critically endangered status, which is a UN-based conservation organization that does like a barometer of life on the whole world. And that was our first pass. That happened a couple of years ago. And just last month, actually, NOAA, the National Oceanic and Atmospheric Administration, took a petition that I helped contribute to, and they have now proposed them as threatened listing in the United States. So they're going to be under the Endangered Species Act here in the next probably year or so. They won't be endangered, they'll be threatened, which is a little good, but not great for them in general. Okay, so where are they? As we said a little bit earlier, they got wiped out in much of the contiguous US. The purple is where they used to be. So they used to be all purple, right? They used to be everywhere. They were all over the place. And back in, and after the disease, what we see is black, 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 like literally not one. And as you get up into the Salish Sea, we start seeing a couple little purples, some hot spots left in the fjords up in British Columbia, some hot spots left up in Prince William Sound, but other than that, it's really patchy and not too many made it through. So I'm sure you all want to know what drove this. Why is this happening? What happened here? So we did an analysis, and I'm not going to get into details, but essentially the graph shows how important is a given factor in determining whether you're going to see a sea star or not. And this purple stuff, the purple bars, are before wasting event. So in the purple, we can compare temperature, depth, chlorophyll, which is phytoplankton, like how much food's in the water. Phytoplankton food, not sea star food. And salinity, so saltiness, and substrate, so sand, rock, clay, whatever. And what you can see is that like before wasting, they did not care. They were like, I'll live there, and I'll live there, and I'll live there too. And they only cared about depth. So they were found in the shallows and not too much in the deeps. They like dropped off around 100 meters. But after wasting in the red is their current predictions of what drives them. And what we can see is that depth dropped down and temperature dropped way up. Or it came way up, right? So the importance of temperature for whether you're finding a sea star or not increased by 450%. So that's a huge difference. And everyone's probably thinking, what does this have to do with climate change, right? Well, the climate change is probably not the trigger. It's probably not why it happened in the first place. It probably was going to happen anyway. Diseases, COVID example, exhibit A, right? Like just kind of happen sometimes. However, what is very clear is that warmer water increases the severity. It makes it spread faster. It makes them die faster and makes more of them die. And it caused that northward range shift. Okay, So it's not necessarily changing climate that caused it, but it's not helping at all. And what this means for us in Oregon, right, where we don't have any sea stars left, is that recovery is a lot harder. So these little guys that are trying to come back are coming back to a system that is warmer than it used to be, and they're more vulnerable than they used to be. So what are we going to do about it? We have formed the roadmap to recovery for the sunflower sea star. We just published this a couple of months ago. It is like a 50-person consortium of people all trying to work on solving this problem. It's funded by the Nature Conservancy, and we have like people all over the world, 3 countries working on it. But first, I need you. I need your involvement. I need you to connect with your community below. If anyone sees one of these things, I need to know. We have I think 8 confirmed sightings in Oregon now, which is better than it was 2 years ago. So they're out there, they're coming back. But if you've seen this sea star, don't pick it up, leave it there. But take a picture of it if you can, and record your location and your date, and send the picture to seastarwasting.org, that thing that's on your bookmark, okay? Because that is our way of mobilizing the masses to try and figure out when these guys are coming back and where they live. You can also submit it to iNaturalist, which is an app on your phone. But if you don't know about it, don't worry about it. Okay, we're gonna practice. How many sea stars are in this photo? Fingers up. Okay, yell it out. 4, you're right. 4 of them are in this photo. They are pretty, actually kind of camouflaged, right? They look very bright when they're out of the water, but when they're back on the rocks and mottled, they hide pretty well. So this is a group of sea stars that made it through. They were one of the first remnant populations that we found up in the fjords of British Columbia on tribal land. So this was taken by the tribal scientists and sent to me with great fanfare. So what can we do about it? So many of you might have heard that we're actually breeding them in captivity. So Friday Harbor has successfully bred them. They have little guys that are about yay now. They're 3 years old. And Hatfield is actually working on trying to get them here pretty soon. We're also working on the disease itself. We have to have a test, right? We can't imagine trying to control COVID without a COVID test. Right? We need a test. We need to know what that disease is before we can move things around very easily. And then some of the things that I'm personally working on are tracking growth. Before this, we actually didn't know how old they were when they were a given size. Like we didn't know whether this was 5 years, 10 years, 15 years, no idea. And so the one in my hand's about 2 years old. It's a group of sea stars I've been tracking for 3 years up in Whidbey Island. And we're doing things like measuring their growth rates and measuring their reproduction. We want to know when they start making eggs. And then how many eggs do they make every year? And how many eggs do they make when they're this big or that big, right? Because that tells us how fast their population might recover. And we're considering reintroductions, like I said a moment ago. ODFW, Hatfield, Aquarium, we're all talking, and we're trying to figure out how to get them back in Oregon waters in the next year, 2 years, something like that. All right, so we are getting close to the end. And I am going to ask you what sunflower sea stars eat. Some sea stars eat other sea stars, not sunflower sea stars. You're thinking of the Dawson star, but correct in some ways, yes. Sea urchins, they love eating sea urchins. So this is a shot of one that just captured a poor little sea urchin. It is engulfing it, and it's going to swallow it, digest it whole, and spit the test out, or the shell out in another day or so. It is terrifying, isn't it? And this is what it looks like when you find the one Californian sunflower star that we know of in the whole world. So if you see it, shout out. Where is it? Where is it? Where is it? There. Ah! And you'll see the diver go like this in just a second because he is so stoked. Oh, wait, I must have cut that part off. Okay, so what you can see, though, is it's got its arms up and it's attacking that sea urchin, those sea urchins, and those sea urchins are skedaddling. This is called an urchin stampede. And it actually has at least 2 in its guts right now. And we're going to rewind the video and go backwards. And I want you to notice, now that you know where the sea star is, can you tell where it's been? That whole section is where it's been. So a sea star can eat about half an urchin a day, but it can scare thousands. And that's what I'm working on this summer in Alaska. We're trying to understand how sea urchins are not only eaten by, but scared by sea stars, because we are really interested in what sea urchins eat. What do sea urchins eat? Kelp. So sea urchins eat kelp, and our Oregon, especially our Oregon kelp forests, have been on the decline, right? They've been getting walloped by these sea urchins. They form what's called a barren. This is kind of the opposite of a stampede. They're coming forward now and eating this kelp. And this is real bad news for kelp, because sea stars died back in 2013-ish, followed by a boom of baby sea urchins because there are no predators left. And we actually have mechanistic links about that now. And then the marine heat wave, the blob, came in and wiped the kelp out. And the kelp are like tomatoes. They grow back every year from spores. They're not like trees. So the fact that there aren't that many left means there are not that many babies being made. And the urchins keep going up and the kelp keep going down. And we're at a state now on the Oregon coast where we've got lots of urchins and not a lot of kelp. And that's important because kelp help you, right? They sequester carbon and mitigate climate change. They're kind of like trees. They feed and nurture all sorts of important marine organisms, just like a forest. Things live in it. And they're better than trees because you can eat them. You and me can eat them. All sorts of things can eat them. Not a lot of stuff eats trees. So they support an entire ecosystem, and that supports things that we like to eat. So fish, abalone, sea urchins, and crab all use the kelp forest, at least at part of their life cycle. So the Oregon Kelp Alliance is all about helping the kelp, and we need to help the kelp right now. So we want the sunflower sea stars back for sure. But in the meantime, we got to get rid of these urchins, right? And I'm not saying urchins are bad. Don't go smashing urchins, please. Unless I tell you to. So we have certain spots along the coast where we have urchin culling experiments, and divers can come help and come down, and it's pretty darn fun. Even though it's morbid, we smash urchins with hammers. But what we do is we create these little kelp oases so that the baby kelp can grow back and recover in that one spot. The other thing we do is we monitor these sites with divers. So if you're a scuba diver and you want to help, we have a whole training program. We'll help teach you how to go count all the stuff in the kelp forest. You can help do that through reef check surveys. We're also partnering with lots of community members. So Oregon Sea Farms is taking those urchins, instead of smashing them, and fattening them up with dulse, with algae, and selling them to restaurants. And we're trying to get y'all to start eating uni because we need to start eating some of these sea urchins to get rid of them. We're also working with dive outfits and whale watching tours. They actually are the boats we use on our dives because they want to get the kelp forest back for their tourism industry too. So these are all the studies we have going on in the Oregon coast right now. It's going to go really fast, but what you can see is we're everywhere. There's people working all over the coast just this summer to analyze and understand kelp forests using tuba, drones, underwater ROVs, satellites, all sorts of stuff. And the Orca group is it's like, I don't know, 50 people or something, trying to work on all aspects of kelp forest health. All right, so I'm going to skip a bunch of stuff because we're not going to get to it. And the only thing I want to say is I'm going to have these guys go hand out their magnets. So if you ever see something strange happen out in the intertidal, like you're poking around on the beach and you see something dead or something dying, I want you all to Google this thing called Primed Network, okay? And there's a way to go in there and sign up and record your sightings and send them to us using an app on your phone. It's called iNaturalist. Has anyone used iNaturalist before? Oh, good. If you haven't, go download it and fool around with it. It's like the coolest thing ever. It'll blow your mind. You can take a picture of your kid and it'll be like Homo sapien. And then you can take a picture of your dog and it'll be like canine, whatever dogs are, lupus, something or other. And then you can take a picture of algae and it'll be like dead man's fingers. And so it tells you what the species is, and you can log it into our marine disease project. It's called Marine Wildlife Health. And you can say it's healthy or it's diseased and send it to us. There's all sorts of tutorials on the website, all sorts of ways, but essentially all you need is a phone and some spare time, and you can help us be our eyes and ears out on the ocean and help us be primed for the future so that we can be more reactive to the next disease that comes by, or proactive when the next disease comes by rather than reactive. Okay, so how to get connected with your community below. Follow the Oregon Kelp Alliance or at PISCO Science on various social media accounts. Report any sunflower star sightings, please, to seastarwasting.org. If you're a diver, go dive with Reef Check, or you could even get in on the urchin smashing if you really want to. There's more red tape. And then you can go to a BioBlitz. Those happen every summer. They're really fun. You can report species that you see, whether they're healthy or not, to that Marine Wildlife Health Project on iNaturalist. You can eat some uni, challenge yourself, please do, and go fishing, and you can go tide pool. So please, if you have any questions, go ahead and email me at my emails, which is up here, and thank you so much. Okay. Oh, I'm not going to touch that.
All right. So we're going to open it up to some questions. We've got a little bit of time. Shannon, if there's anybody online with questions, go ahead and raise your hand. For folks in the room, you can either go to that mic or you can raise your hand and I will bring it to you. And I see the first one is at the very back of the room. So here I go. Oh, did you guys get the... Oh yeah, pass out those stickers, boys. Dr. Gravem, if you can't hear the mic, you might have to step in front of the speaker that's up on the ceiling. Look straight up.
Do you want me to just do this?
No, we can hear you just fine. You can't hear us.
Oh, okay.
So when you were showing the population graph of the sunflower sea stars, I noticed a steep decline at like 2010, 2011.
Yeah.
Which that was before the sea star wasting disease. What was that attributed to?
It was people not taking data on sea stars. So before sea star wasting disease, these things were pretty common. And we didn't have like like concerted sampling effort happening, especially up in Alaska where they were really, really abundant. And so it kind of looks like there's a pre-wasting drop, or at least like they're low and they go up. And the up, at the very least, is due to us going, oh my gosh, this disease is coming. Everybody go out and count them before they die. So I don't think that they did bump around, but I don't think that pre-wasting divot is real. Yeah. Thanks. Good catch.
All right. Other questions?
So wouldn't you expect the sea urchin population to take a dive if the kelp forest is really dropped as precipitously as it has?
Yeah, eventually they probably will take a dive, and sea urchins are notoriously prone to disease as well. So we may end up with the sea urchin disease, who knows? But sea urchins are very good at starving. And so they are essentially just a shell and a gut and some eggs. That's all that's to them. And so they can just wait it out. They'll eat, like they'll scrape the algae that grows just kind of as a scuzz on the bottom. They'll do that. They'll eat plastic. They'll eat transect tapes that cost $50 a pop. So they're really good at just kind of hanging out in a zombie state. for a long, long time, and they don't really die from starvation without, like, you have to starve them for like 6 months straight, no food. And that doesn't really happen in the ocean. There's always something that they can eat. Plastic. Yeah. I don't think it's very nutrient rich, but
I have two questions. My first one is the increase in urchin population a result of the decrease in the predator population? And then the second question is, when we're talking about reintroducing the sea stars back into the ocean, what are your expectations for how well they'll survive
Yeah.
in these new conditions?
Yeah. So the first question was whether we think that the sea urchin increase was actually due to the predator decrease. And we've been arguing about this a lot. The coincidence is uncanny, right? Like, everyone talks about otters being really important for urchin populations, but we haven't had otters here for 100 years. And the kelp were doing just fine, and the urchins were, not super abundant. And then the sea stars were gone for a year or two, and the urchins went bananas, right? So first of all, like, the coincidence and timing, and it's like consistent between places, is really striking. And then just recently, like 2 months ago, we finally got the data to actually use like a model and mathematically calculate, if a sea urchin eats this much and there used to be this many sea stars, could they keep sea urchin populations down? And the answer was almost invariably yes. So we now think that there's like a pretty strong link between the urchin increase and the sea star decline. But that's one thing that we're going to keep working on, because not everyone's convinced yet, but they will be after I'm done with them. Yeah.
All right.
Oh, second question. She had a second question.
Second question.
What was it? Oh, how will they do? Right. How will they do on reintroduction? We're not quite sure. We're taking baby steps, right? So right now they're still in Whidbey Island, Salish Sea, Seattle area, and they're doing okay up there. And so we think Oregon is a great baby step. We're going to try and get them down here because we have cool enough water. We have plenty of sea urchins for them to eat. And we think that as long as the disease doesn't come back in a big way, that they would be okay. We're certainly not going to go like try and put them in Mexico right away, but maybe someday. We don't know. There is evidence that there has actually been evolution in the populations since wasting. Like, it's been so strong, it's caused their DNA to change. And so we don't know if it's actually they're more resistant or not. We just know that they've changed. And so hopefully that means they've changed for the better and they're stronger now.
All right, we're going to take one more question at the mic stand. That mic is turned off. There's a slider on the top that if you slide it up, the mic will go live.
Okay, lovely. Sorry. I got here a bit late, so you might have covered this already, but what organisms in particular benefit from these kelp forest ecosystems?
Everything. Humans, for one. But really, abalone is a big one. They eat kelp, and there's not a lot of kelp, so the abalone are starving. And urchins themselves benefit from the kelp, even though right now they're just wandering around like. And then rockfish really like to recruit to kelp. So baby rockfish live their first couple months in kelp. And so without kelp, there's nowhere for them to settle and they get gobbled up. And then crabs like to recruit to kelp. And so anyway, there's all sorts of like upstream effects, and they're the most prolific seaweed. So it's kind of like everything benefits from grass, right? Like, they're the base of a lot of food chains.
Right.
Was that your question?
Yeah.
Cool. I have a sad story. You want to hear it?
You want to end our talk on a
Yeah.
sad story?
No, I don't. No, I don't. No, I don't. No, I do have to. I have to now. So I was diving with, I think you, Caroline, in Port Orford, or snorkeling. And we were down there looking for the last kelp blade in Port Orford. There was one. It had been there for a whole year. It was decrepit. And I go down to the bottom of it, and there are like 50 urchins, like, and there was one abalone licking it. And the urchins were like on its back, and it was going, and trying to eat the kelp. It was so sad. But that spot is the place we cleared the urchins out of. And the abilene are doing good there. So it's going to be okay.
See, it's a good story.
Yeah.
All right, everybody. Thank you so much for being here. Thank our Sarah.
Thank you, my assistants.
Thank you, assistants.
Thank you, assistants.
And remember, if you want to learn a little bit more, we'll do a Science on Tap around the sea urchin topics here with Tom Calvanese, which was on one of the slides, on the 26th of this month. Go have fun at Marine Science Day.
Yeah.
Awesome. Thank you all for being here.
All right. Did it. Did it. Oh, yeah.
Thank you for that. For folks online, we're going to end the presentation now. Thank you very much for being here.